Cell, the unit of life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism live? Or what is it that an inanimate thing does not have which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed of cells. Some are composed of single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of one independent existence and to performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Schleiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodore Swan, 1839, a British biologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cell. On the basis of this, Swan proposed the hypothesis that the bodies of animal and plant are composed of cells and product of cells. Schleiden and Swan together formulated the cell theory. This theory, however, did not explain as to how new cells are formed. Rudolf Virchow, 1855, first explained the cell divided and new cells are formed from pre-existing cells. He modified the hypothesis of Schleiden and Swan to give the cell theory a final shape. Cell theory, as understood today, is 1. All living organisms are composed of cells and product of cells. All cells arises from pre-existing cells. An overview of cell. You have earlier observed cells in an onion peel and or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cell of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosome, which in turn contains the genetic material, DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bounded distinct structure called organelles like Endoplasmic Reticulum, the Golgi complex, lysosome, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in cytoplasm but also within the two organelles, chloroplast in plant and mitochondria, and on rough ear. Animal cells contain another non-membrane bound organelle called centrosome, which helps in cell division. Cell differs greatly in size, shape, activity. For example, mycoplasm, the smallest cell, are only 0.3 micrometer and length, while the could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasm, and PPLO, pneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shapes and size. The four basic shapes of the bacteria are Bacillus, Coccus, Vibrio, and Spirula. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane, except in mycoplasma. The fluid matrix filling the cell is cytoplasm. There is no well-defined nucleus. The genetic materials is basically naked, not enveloped by a nuclear membrane. In addition to genomic DNA, the single chromosome or circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmid. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher class, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organella like the one in eukaryotes are found in prokaryotic cell except for ribosome. Prokaryotes have something unique in the form of inclusion, a specialized differentiated form of cell membrane called mesosome is the characteristics of prokaryotes. They are essentially enfolding of cell membrane. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is outermost glycolates, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the difference in the cell envelope and the manner in which they respond to the staining procedure developed by Crumb. Is those that take up The gram-stain are gram-positive and the others that do not are called gram-negative bacteria. Glycocalyx differ in composition and thickness among different bacteria. It could be a loose sheet called the slime layer in some, while in others it may be thick and tough called capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacteria from bursting or collapsing. The plasma membrane is selectively permeable in nature and interact with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by extension of plasma membrane into the cell. These extensions are in the form of vesicle, tubules and lamella. They help in cell wall formation, DNA replication and distribution to daughter cells. They also help in respiration, secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, villi and fimbriae are also surface structure of the bacteria but do not play a role in motility. The villi are elongated tubular structure made a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rock in stream and also to the host tissues. Ribosome and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. The ribosome of a polysome translates the mRNA into proteins. Inclusion bodies. Reserved materials in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example: phosphate granules, cyanophysin granules and glycogen granules. Gas vacuole are found in blue, green and purple and green photosynthetic bacteria. Eukaryotic cell. The eukaryotes include all the protists, plants, animal and fungi. In eukaryotic cells, there is extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possesses cell wall, plastic, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are present in almost all plant cells. Let us now look at individual cell organelles to understand their structure and functions. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipid and protein. The major lipids are phospholipids that are arranged in bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and hydrophobic tails towards the inner part. This ensures that the nonpolar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to phospholipid, membrane also contain cholesterol. Later, biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of proteins and lipids varies considerably in different cell types. In human beings, the membrane of the erythrocyte has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lie on the surface of membrane, while the integral proteins are partially or totally buried in the membrane. An improved model of structure of cell membrane was proposed by Singer and Nicholson 1972, widely accepted as fluid Mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of the protein within the overall layer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. On the most important function of the plasma membrane is the transport of the molecule across it. The membrane is selectively permeable to some molecule present on either side of it. Many molecules can move quickly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solute may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecule cannot pass through the nonpolar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to higher concentration. Such transport is an energy dependent process in which ATP is utilized and is called active transport. Example, sodium ion, potassium ion pump. Cell wall. As you may recall, a non-living rigid structure called the cell wall forms an outer covering of the plasma membrane of fungi and plant. Cell wall not only gives shape to the cell and protect the cell from the mechanical damage and infection, it also helps in cell-to-cell interaction and provide barrier to unbearable macromolecules, as we have cell wall made of cellulose, gelatin, mannans and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectin and protein. The cell wall of a young plant cell, the primary wall is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is formed on the inner porous membrane side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cell. Endomembrane system. While each of membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome and vacuole. Since the function of the mitochondria, chloroplast and peroxisomes are not coordinated with the above component, these are not considered as part of the endomembrane system. The endoplasmic reticulum. Here, electron microscopic studies of eukaryotic cell reveal the presence of a network or reticulum of tiny tubular structure scattered in the cytoplasm that is called the endoplasmic reticulum. Hence, here divides the intercellular space into two distinct compartments, that is luminal inner side here and extraluminal cytoplasm compartment. The ER often shows ribosome attached to their outer surface. The endoplasmic reticulum bearing ribosome on their surface is called rough endoplasmic reticulum. In the absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum. RER, rough endoplasmic reticulum, is frequently observed in the cell wall activity involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in ACR. Golgi apparatus. Camillo Golgi, 1898, first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat disc-shaped sacs or cisterns of 0.5 micrometer to 1 0.0 micrometer diameter. These are stacked parallel to each other. Varied number of cisterna are present in Golgi complex. The Golgi cisterns are concentrically arranged near the nucleus with distinct convex CIS or the forming phase and concave turns or the maturing phase. The cis and the trans phase of the organelles are entirely different but interconnected. The Golgi apparatus principally perform the function of packing materials to be delivered either to the intracellular target or secreted outside the cell. Material to be packed in the form of vesicle from the ear fused with the cis phase of the Golgi apparatus and moved towards the maturing phase. This explains why the Golgi apparatus remains in close association with the Endoplasmic Reticulum. A number of proteins synthesized by ribosomes on the Endoplasmic Reticulum are modified in the cistern of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formation of glycoprotein and glycolipids. Lysosome. These are membrane-bound vesicular structure formed by the process of packing in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolysases, lipase, proteinase, carbohydrates, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrate, protein, lipids, and nucleic acid. Vacuoles. The vacuoles is the membrane-bound piece formed in the cytoplasm. It contains water, sap, excretory product, and other materials not useful for cell. The vacuole is bound by a single membrane called tonoplast. In plant cell, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of the number of ions and excretes other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particle. Mitochondria. Mitochondria, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cell. In terms of the shape and size also, considerable degree of variability is observed. Typically, it is sausage shape or cylindrical, having a diameter of 0.2 to 1.0 micrometer, average 0.5 micrometer, and length 1.0 to 4.1 micrometer. Each mitochondria is a double membrane-bound structure with the outer membrane and the inner membrane, dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infolding called the cystia. Cristia. Towards the matrix, the cristia increases the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called powerhouse of the cell. The matrix also possesses single circular DNA molecules, a few RNA molecules, ribosomes 70S, and the component required for synthesis of protein. The mitochondria divide by fission. Plastid. Plastid are found in all plant cells and euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigment, thus imparting specific color to the plant. Based on the type of pigment, plastid can be classified into chloroplast, chromoplast, leucoplast. The chloroplast contain chlorophyll and carotenoid pigment, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigment like carotene, xanthophylls, and other also present. This gives the part of the plant a yellow, orange, or red color. The leucoplast are the colorless plastid of varied shape and size which stored nutrient. Aminoplasts store carbohydrate, starch, example potato. Eleoplast store oil and fat, whereas the eleoplast store protein. Majority of the chloroplast of the green plants are found in the mesophyll cell of the leaf. These are the lens-shaped, oval, spherical, discoid, or even ribbon-like organelle having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from one part cell of the chlamydomonas, green algae, to 20 to 40 part cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized patent membranous sacs called the thylakoid are present in the stroma. Thylakoids are arranged in stack like the piles of coins called grana or the integral thylakoids. In addition, there are flat membranous tubules called the stroma lamella connecting the thylakoids of the different grana. The membrane of the thylakoid enclosed a space called a lumen. The stroma of the chloroplast contain enzymes required for the synthesis of carbohydrate and protein. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller 70S than the cytoplasmic ribosomes 80S. Ribosomes are granular structure first observed under the electron microscope as dense particles by George Pallet 1953. They are composed of ribonucleic acid, RNA, and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. Each ribosome has two subunits, larger and smaller subunit. The two subunits of 80S ribosomes are 60S and 40S, while that of 70S ribosomes are 50S and 30S. Here is Bedbugs unit stand for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits: cytoskeleton, an elaborate network of filamentous proteinaceous structure consisting microtubule, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of cell. Cilia and flagella. Cilia and flagella are hair-like outgrowth of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possesses flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscope study of a cilium or the flagellum shows that they are covered with plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonemal microtubules is referred to as the 9+2 array. The central tubules are connected by bridge and is also employed by central sheath, which is connected to the one of the tubules of each peripheral doublet by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal body. Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structure called centrioles. They are surrounded by atmospheric pericentriolar materials. Both the centrioles in the centrosome lie perpendicular to each other, in which each has an organization like cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin proteins. Each of the peripheral fibrils is a triplet. The adjacent triplets are also linked. The central parts of the proximal region of the centrioles is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles from the basal body of the cilia or flagella, the spindle fiber that give rise to the spindle apparatus during the cell division in animal cell. Nucleus. Nucleus as a cell organelle was first described by Robert Bound as early in 1831. Later, the materials of the nucleus stained by the basic dye was given by the name chromatin by Fleming. The interphase nucleus of a cell, when it is not dividing, has highly extended and elaborate nucleoprotein fiber called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer called perinuclear phase, form a barrier between the material present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosome on it. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are the passage through which movement of RNA and protein molecule takes place in both directions between the nucleus and the cytoplasm. Normally there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect names of the organisms that have more than one nucleus per cell? Some mature cells even lack nucleus. Example, erythrocyte of many mammals and seed tube cells of vacuolar plants. Would you consider these cells as living? The nuclear matrix or nucleoplasm contain nucleus, nucleolus and chromatin. The nucleoli are the spherical structure present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm, as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Large and more numerous nuclei are present in cell actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. During different stages of cell division, cell shows structure chromosome in place of nucleus. Chromatin contains DNA and some basic proteins called histone, some non-histone proteins and also RNA. A single human cell has approximately 2 meter long thread of DNA distributed among its 46 23 pairs chromosomes. We will study the detailed DNA packaging in the form of chromosome in class 12. Even chromosome visible only in dividing cell essentially has a primary constriction or the symptom on the side of the D-shaped structure called kinetochores are present. Centromere hold two chromatin of the chromosome. Based on the position of the centromere, the chromosomes can be classified into 4 types. The metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming the extremely short and one very long arm, whereas the telocentric chromosome has a terminal chromosome, terminal centromere. Sometimes a few chromosomes has non-staining secondary constriction at a constant location. This gives the appearance of small fragment called a satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plants and animal cells.